Hello and welcome back. We're going to continue going over the hypersensitivities with this video. We're going to focus on this video on the type 2 hypersensitivity. Okay. Now with the type 2 hypersensitivity, the antibodies that tend to be involved in this are IgG or IgM. Okay. As compared to the type 1, which was IgE. All right, so there's a different antibody that's going to work here. Right? So, and what the type 2 hypersensitivity is going to do, this one is going to target cells. Okay, and typically what you have is you have the antibody binding to a protein most often on the cell. Okay, so if I go ahead, this is our friendly neighborhood cell here. Okay, on the outside of it, there's going to be some protein. All right. And then what happens is the body makes an antibody that recognizes that particular protein. Okay? So the antibody I've drawn here is IgG just because it's a little simpler for me to draw. But this could be an IgM antibody as well. And what that typically does then is the let me draw that in. The part of the antibody that's sticking out is called the Fc portion. You should maybe remember this from microbiology. But what that does is activate a protein or series of proteins in the blood known as complement. All right, those complement proteins will recognize the Fc portion. And then what they do is that complement works to essentially open up a pore in the cell, and the cell ends up being lysed if complement is the way this is activated. Okay, so complement is one way this can happen. The other way that this can happen is that antibody can target the cell to be taken in by phagocytosis. All right. Either of these are ways that that can happen. All right. But what you've got is something on the cell that's recognized as foreign. There are antibodies that already exist that recognize this. All right. Some examples of type 2 hypersensitivities are transfusion reactions. Okay. So if somebody has type A blood, what that means is they make antibodies to type B, okay, and also to type AB, all right? So you have that antibody that's there and is present. So if somebody who has type A blood is incorrectly given type B blood, okay, that's going to trigger this type 2 hypersensitivity, and the body's going to destroy those red blood cells that were transfused in because the individual's body is recognizing that those red blood cells as being foreign. Okay, because there's something on the outside of the cell that isn't what is recognized as self. okay? So that's one example. I'll put a 1 there. Our second example is the hemolytic disease of the newborn, okay? Now, this is an issue where mom and baby have different blood types, or more commonly, different Rh factors, okay? So the Rh factor could also be what stimulates this up here with a transfusion reaction. So if somebody is type A negative and they get type A positive blood, that's also going to stimulate that transfusion reaction, okay? With the hemolytic disease, the Rh factor incompatibility is typically what triggers this, okay? And in this case, mom is always Rh negative. Baby inherited an Rh positive. From the dad, okay? So what ends up happening with the hemolytic disease of the newborn, or the Rh incompatibility here, the mom will develop antibodies to the Rh factor on the outside of the baby's blood, because with the placenta, there's some degree of mixing between mom and baby that can happen. There is also mixing of mom and baby's blood that can happen during delivery. In the case of hemolytic disease of the newborn, all right, mom first has to develop the antibodies though. So mom's first pregnancy with an Rh positive baby typically goes okay. But subsequent pregnancies where baby is Rh positive, the mom can develop antibodies to that Rh factor, which then recognize the developing fetus as foreign. There is a way around this. There is a shot that moms who are Rh negative, so if they have O negative, A negative, B negative, or AB negative blood, they're given RhoGAM typically around mid-pregnancy, and then at delivery. Because what that RhoGAM does is that RhoGAM is essentially antibodies to the Rh factor, and those antibodies destroy the red blood cells so that mom doesn't actually make the antibodies, okay? Essentially keeping her immune system from recognizing the Rh factor as being foreign, okay? So treatment with this, if mom is Rh negative, and dad has Rh positive blood, this is typically standard treatment for that mom during her pregnancy, during the first pregnancy and all subsequent pregnancies, okay? So that's a second example behind the transfusion reactions. Third example that I've got for you associated with a type 2 hypersensitivity is myasthenia gravis, okay? This is an autoimmune disease. And what happens with myasthenia gravis is the individual develops antibodies to the acetylcholine receptor, okay? And so acetylcholine receptors are found on the plasma membrane of muscle cells, right? Because if you remember from anatomy and physiology, acetylcholine released at the neuromuscular junction that's going to stimulate muscle contraction. Right? But if you're sitting there on the plasma membrane, and let me put in our acetylcholine receptor here as kind of this pink blob, right? This is going to be our acetylcholine receptor. Up here, this would be our neuron associated with our neuromuscular junction, okay? Right? When that action potential signals the neuron, right, it releases the acetylcholine into that neuromuscular junction, into that synapse, where the acetylcholine would bind to the acetylcholine receptor, and you get muscle contraction, right? But if I erase this over here, let me put another acetylcholine receptor right here, right? We've got an acetylcholine receptor. For individuals who have myasthenia gravis, what ends up happening, they've got an antibody that's going to bind to that receptor, all right? If the antibody is bound to the receptor, acetylcholine can't bind to the receptor. So essentially, that's how that interferes with the muscle contraction, all right? So in this case, we aren't necessarily getting the cell to lyse open like we did with the transfusion reaction and the hemolytic disease of the newborn, where the red blood cells are lysing. This one is essentially blocking the receptor, okay? Similar kind of thing can happen. I've run out of board space, but I don't want to make one more video to put up my other example, which is Graves' disease, okay? Graves' disease is also an autoimmune disease. Graves' disease attacks the thyroid in the neck, all right? So essentially, it's a similar kind of thing. There's an antibody that attacks the thyroid stimulating hormone receptor, and what ends up happening when it binds that receptor, you get too much thyroid hormone produced, and essentially what happens, the thyroid burns out, okay? So myasthenia gravis and Graves' disease are similar in that you've got an antibody that's binding to a receptor. It's different in that myasthenia gravis is interfering with the signaling process at the neuromuscular junction, whereas with Graves' disease, it's actually stimulating the thyroid cell to release lots of thyroid hormone, okay? But these are type 2 hypersensitivities, either IgG or IgM mediated. Complement can be either IgG or IgM. The myasthenia gravis and the Graves' disease, that tends to be an IgG mediated hypersensitivity. That kind of sums up the type 2. If you have questions, please let me know.